My dear students, in this class, let us try to learn about the embryonic development of the frog until organogenesis. Frog is an amphibian animal. It belongs to the class Amphibia and it exhibits indirect development. Indirect development means this is the life cycle that includes larval stages. In case of life history of frog, there is one specific larva called tadpole larva, which is physically, physiologically, biochemically different from the adult frog. If the larva is transformed into adult, that particular process is known as metamorphosis. So, like that, the metamorphosis in case of frog is highly complicated and it includes a developmental stage called a tadpole larva, and before that there are series of developmental stages. And this development of frog includes gametogenesis, fertilization, formation of zygote, cleavage, morula, blastula, gastrula and organogenesis. Until gastrulation, in order to form the gastrula, the various cells that are present in the embryonic stages will undergo a specific migration, specific movement, and these cellular movements for the purpose of gastrulation are called morphogenetic movements. So, like that, the frog developmental stage includes various morphogenetic movements and the formation of germinal layers. Once the germinal layers are formed, each germinal layer will give rise to a particular system. So, like that, organogenesis will come into play after the gastrulation. So, let us discuss one by one all these components. Frog belongs to the phylum Chordata, subphylum Vertebrata, superclass Gnathostomata and class Amphibia. Amphibia includes those animals which lead dual life, which lead amphibious life. Exactly this aspect has made the development of frog a complicated process. Because the tadpole larva is aquatic in nature, it is strictly aquatic, whereas the adult frog is amphibious. So, if you observe these developmental stages in the frog, it clearly indicates a shift from aquatic environment to terrestrial environment. So, there is a very clear cut shift, very clear cut choice, a gradual affinity from aquatic mode to terrestrial mode of life. So, that is the reason why the tadpole larva frog is aquatic and the adult frog is amphibious. That means it is about to invade the terrestrial environment in the future evolution process. And it belongs to the subclass Lysamphibia and order Anura. It exhibits indirect development and the larval stage is called tadpole larva, which contains tail. Transformation of this tadpole larva into adult frog is called metamorphosis. And here you can see there is a clear cut demarcation, clear cut contrast between tadpole larva frog and adult frog. The tadpole larva is aquatic, whereas adult frog is amphibious, and the tadpole larva has tail, whereas adult frog is devoid of tail. During the process of development, certain proteolytic enzymes are secreted by this developmental stages, which will absorb the tail bud, which will absorb the tail region. And the tadpole larva is gill breathing because it is aquatic in its habitat. And whereas adult frog being amphibious in nature, it has shifted to lung breathing habit. And the tadpole larva feeds on plant material, that means it is a herbivore, whereas adult frog is entomophagous, that is it is insectivorous. So, like that, here you can see the aquatic tail containing gill breathing herbivorous larva, tadpole larva, is gradually metamorphosed into amphibious tailless lung breathing insectivorous adult frog. So, this is exactly called metamorphosis. Just you can imagine what is the magnitude of this metamorphosis, how much of adjustments might have taken place inside the embryo to transform this larva into the adult frog. And here there are many developmental stages that are seen in the frog life history, namely gametes. Male gametes and female gametes are released into the water because we know that amphibians go back to water for the purpose of reproduction and for the purpose of fertilization. Because in case of frog there is no copulatory organ, only pseudocopulation occurs. This pseudocopulation is called amplexus. And during the process of amplexus, egg moss and spermatozoa both are released into the water medium. That means the fertilization is external, it occurs in the water. And due to fertilization, a single cellular structure, unicellular structure, namely zygote is formed, and this zygote undergoes cleavage. And subsequently, morula stage, blastula stage and gastrula stage are formed, followed by the organogenesis. Development of different organ systems is called organogenesis. And in this process, primary germinal layers are formed, namely ectoderm, mesoderm and endoderm. And all these primary germinal layers will give rise to different sets of organ systems. Here you can see on the right side the complete developmental cycle of the frog. The adult frogs, both male and female, during the process of amplexus, amplexus is nothing but pseudocopulation. During the process of amplexus, they release spermatozoa and ova in greater numbers. Normally these are called as spawn and milt. When they are released into the water medium, they come together just by chance, accidentally. It's a very sweet, lovable coincidence. And when the spermatozoa reaches the egg, it fertilizes the egg, forming the zygote. Here you can see the zygote has undergone cleavage, and you remember cleavage is nothing but the mitotic divisions that are exhibited by the zygote. Cleavage divisions are always mitotic, and the ball of cells is formed, and that is called morula. Later blastula is formed, and ultimately gastrula is formed. And here in the further developmental stages, you can also see the formation of different organs like neural plate, neural tube, notochord, nerve cord, like that. Ultimately, tadpole larva is hatched out of the egg. Later, it is gradually metamorphosed into the adult frog. And here, while dealing with the egg of the frog, we come across one term called grey crescent. This grey crescent corresponds to the future dorsal or back side of the embryo, while the sperm entry site corresponds to the ventral or belly site. That means grey crescent corresponds to the future dorsal site. And exactly on the opposite side of the grey crescent, the spermatozoa will enter into the egg. The grey crescent marks the future dorsal side of the frog. So here you can see the different developmental stages of the life history of frog. The movement of cells during the embryonic stages to form a clear-cut gastrula is known as morphogenetic movements. And during gastrulation, various morphogenetic movements like invagination, involution, ingression, epiboly, intercalation and convergent extensions are seen. Invagination, of course, is the initial step of gastrulation. In involution, a tissue sheath rolls inward, and splitting of one sheath into many layers is called delamination. And thinning of sheets is called epiboly. It results in the increase of cell surface. No doubt during the process of epiboly, the cell is being thinned, but at the same time there is a clear-cut increase in the cellular surface. And here you can see the diagrammatic representation of the different morphogenetic movements. So here you can see the invagination, what is happening. The cells are just going inward. And in ingression, you can see the apical and basal cell, how they migrate with respect to each other. And in involution, how it is taking a turn. Epiboly is nothing but the thinning of the cell, no doubt, but at the same time it is resulting in the expansion or else the increase in the cell surface area. In convergent extension, you can see different layers converge to continue. So that is about various morphogenetic movements like invagination, involution, ingression, delamination and epiboly. And due to this systematic programmed morphogenetic movements, a wonderful structure called gastrula is also formed, and from which due to organogenesis, different organ systems will develop. So anyway, the ovum of frog, that means the egg of frog, is mesolysethal. That means it is loaded with only moderate quantity of yolk material. And this yolk is distributed in such a manner that much of the yolk is concentrated at the lower side of the egg, which is called a vegetal pole, and that is yolk rich region. Whereas there is one more pole at the opposite end of the egg, that is called animal pole, which is yolk poor, and it is non-cladoic. Up to amphibians, you don't find cladoic eggs, because cladoic egg has made its appearance for the first time in reptilia. Cladoic egg is the egg that is surrounded by the calcium carbonate shell, whereas in amphibians you find only non-cladoic egg which is devoid of calcium carbonate shell. Because of the absence of calcium carbonate shell, they have to lay their eggs in the water medium. They have to lay their eggs in the ponds or lakes or any water body like that. Whereas if you take the reptiles which have developed the cladoic egg, because of the presence of calcium carbonate shell, although they lay eggs on the land, they don't undergo desiccation. Whereas here there is a probability of desiccation of the embryo if it is laid on the land. So in order to prevent the desiccation of the embryo that is developing inside the non-cladoic egg, the frogs lay eggs in the water region, that means water bodies. Whatever may be, the fertilization itself is taking place in the water. As a result, eggs are formed in the water and they are also being hatched in the water. So like that, the egg is mesolysethal, telolysethal, non-cladoic egg, and it is divided into animal hemisphere and vegetal hemisphere, yolk poor region and yolk rich region. And the yolk rich region is called vegetal hemisphere. It is always on the base of the egg, because yolk is a very dense material which is attracted towards the gravity. And it is surrounded by plasma membrane, vitelline membrane and jelly coat, of course. Here you can see the various membranes of the egg of frog. And cleavage is holoblastic cleavage, which is complete, and it is unequal cleavage. As a result, megameres, which are big, micromeres, which are small. Two types of cells are formed during the embryonic development, and they exhibit specific movements, because megameres are gravity oriented, whereas micromeres to some extent they conquer the gravity during their mobility. And the cleavage passes through meridional plane and also horizontal plane. And blastula is formed, and this blastula, because of one cavity, it is also called coeloblastula, and this cavity is present in the animal pole region. As a result, such blastula is called eccentric blastula. Eccentric blastula is one which has got eccentric blastocele. Eccentric blastocele is the blastocele that is not formed in the center, that is formed in the animal pole. So that is about the blastula. So here you can see the eccentric blastocele. If the cavity appears at the animal pole, that means if the coelom means cavity, appears in the animal pole, it is called eccentric blastocele. And coming to the fate map is the representation of the fate of blastomeres on the external surface. Region of grey crescent consists of presumptive mesoderm and presumptive notochord. Posterior half consists of presumptive endoderm. That means fate map will clearly demarcate the fate of that particular area, the future potential of that particular cell. So here you can see the fate map where presumptive ectoderm and presumptive endoderm are displayed, and here you can see the neural plate which is going to form the nervous system, and from endoderm digestive organs and lungs are being formed, and this is the vegetal pole and this is the animal pole, and this is the epidermis through which the skin arises, and here you can find the mesoderm from which the muscles, kidney, bone and gonads will arise. Like that, from each layer, from each germinal layer, certain organs are bound to develop. And during the process of gastrulation, morphogenetic movements are very clearly observed. And as I mentioned earlier, there are various types of morphogenetic movements like epiboly, emboli, invagination, and involution. What is invagination means? It is the movement of blastomeres into blastocele by inward folding. This is very important. Movement of blastomeres into blastocele by inward folding is called invagination. And blastomeres enter into blastocele or over the inner surface of the outer ectoderm, that is called involution. That means invagination is only the inward folding, but if this inward folding turns to some side, then that is called involution. That means they enter into blastocele over the inner surface of the outer ectoderm. Epiboly, I told that in epiboly what is happening, the thinning of the cell is taking place, but at the same time there is increase in the surface area. So repeated divisions of micromeres, which are small, push them towards vegetal pole from animal pole. This results in the overlapping of megameres, which are larger, with micromeres except at the region of yolk plug. That means only except at the region of yolk plug, other areas are being overlapped by the micromeres. Formation of blastopore. Formation of blastopore. Here the little behind the edge of grey crescent, archenteron is formed at the later stage and it is represented by crescentic groove. Archenteron is nothing but a primitive gut that is present during embryonic development. And this fate of blastopore is very, very important in the classification of animal kingdom. Because if the blastopore is converted into anus, such animals are called deuterostomes, whereas if the blastopore is converted into mouth, such animals are called protostomes. And frog being vertebrate, it is a deuterostomous animal where the blastopore in the embryonic stages is gradually converted into cloaca or anus in case of adult frog. Because you don't use the term anus in case of frog, you use the term cloaca. And later invagination of endoderm takes place where, as I already told, megameres sink inside, of course leaving the area called yolk plug exposed. And formation of archenteron and blastocele is gradually replaced by the archenteron, which is nothing but the primitive gut. And from the archenteron many pouches arise, thus forming a coelom. And if the coelom is formed due to the pouches of archenteron, such coelom is called enterocele. And here you can see the involution where the three layers, the ectoderm, mesoderm and endoderm are also formed, and these are called primary germ layers. The various organs of the larval body develop from these three layers. What are the three germinal layers means? Ectoderm, this is the first germ layer, endoderm, second germ layer, mesoderm, the third germ layer. And formation of different organs from these three germinal layers is called organogenesis. And let us see what are the different structures, organs, systems that arise from the ectoderm. Ectoderm, as I already told, it is the first germ layer. It forms epidermis of skin and its glands. Pharynx and rectum arise from this ectoderm. Retina, lens, cornea, conjunctiva are ectodermal in origin. Brain and spinal cord are ectodermal in origin. Pituitary gland and medulla of adrenal gland are also ectodermal in origin. And coming to the endoderm, which is the second germinal layer, it gives rise to alimentary canal, liver, pancreas and intestine. Thymus, thyroid and parathyroid glands are endodermal in origin. Respiratory tracts, peritoneum of ureters are endodermal in origin. And middle ear, which is possessed by frog, it arises from the endoderm. And external ears are totally absent in case of frog, of course. And coming to mesoderm, it is the third germinal layer. It forms connective tissue, skeletal tissue like bone and cartilage arise from the mesoderm, and circulatory system and urinogenital system are mesodermal in origin. Adrenal cortex is mesodermal in origin. Sclerotic layer of an eye and coelomic epithelium, myelin sheath of axon are mesodermal in origin. So this is how the various organ systems arise from the different germinal layers of the frog. So this is about the development of frog. Thank you very much.